Welcome to our presentation on mitosis and cytokinesis. The key concept here is the cells divide during mitosis and cytokinesis. We have a cell late in the stages of cytokinesis, the cell, actually the cytoplasm, dividing. Before that can happen, we have to copy the nucleus. There's two parts to mitosis cell division. We have to copy the nucleus, and then we have to copy the cytoplasm, the rest of the cell. Chromosomes will condense at the beginning. Start out with the double helix. There's some DNA and proteins involved, chromatin, and then supercoiled DNA. It coils up and shortens, fattens, thickens, and becomes visible. DNA wraps around the proteins, histones, that condenses it, and we get the classic little X-shaped chromosomes, which are actually that's actually X-shaped chromatids, two identical strands of copied DNA. One half of a duplicated chromosome is a chromatid. Sister chromatids are held together by a centromere, sort of in the center part. They're not exactly in dead center, but they're towards the center of this X. Telomeres protect the DNA, but do not include genes. Chromatid, telomere, and the centromere is where they're held together. Mitosis and cytokinesis produce two genetically identical daughter cells. Copy, copy, copy. Interphase is the normal growth and function of a cell. During interphase, the cell does, if it's going to divide, it prepares to divide by going through the S and G2 phase. S phase is when the DNA replicates. G2 is when the cytoplasmic material replicates, copies. The DNA is copied during interphase, during the S phase of interphase. So we have a nucleus with DNA and the parent cell. Centromeres, spindle fibers begin to form, and the process begins. During prophase, the first phase of mitosis, the phase of nuclear division, chromosomes shorten, fatten, thicken, condense, and spindle fibers form, and the nuclear membrane begins to dissolve. So you can see here the nuclear membrane has a dashed line about it. The centromeres condense, and you're starting to see some fibers or spindle fibers forming. During metaphase, chromosomes line up in the middle of the cell. Spindle fibers help move them by grabbing onto the centromeres and slide them to the center. In this case, notice that they're all lined up like you would maybe in a gym class or a sports team, line up on a line. Every chromosome lined up next to each other. That will be different in meiosis, something that's coming up in the next unit. There's 4 steps to mitosis. First one is prophase. Second one is metaphase. During anaphase, the sister chromatids separate to opposite sides of the cell as the spindle fibers shorten and pull apart the chromatids. We have the spindle fibers shortening, pulling the chromatids, the sister chromatids, the identical DNA strands apart. So the green and the green are 2 identical strands of DNA, the red and the red, 2 identical strands of DNA, and so on. And they're being pulled to opposite sides of the cell. During telophase is pretty much just the opposite of prophase. During telophase, a new nucleus, a nuclear membrane forms. The chromosomes begin to uncoil and stretch out and become more invisible, and the spindle fibers disappear. So you see here we've got the spindle fibers almost gone, the chromosomes are stretching out, and the nuclear membrane is reforming. Now notice at this phase, it's very short-lived, but it's kind of an interesting set. We have one cell with 2 nuclei in it. That's rather strange. And then we need to divide the cell, the cytoplasm. In animal cells, the membrane pinches closed, like squeezing a water balloon. In plant cells, another cell plate or to be a cell wall forms between the two. And there we have the pinching going on in an animal cell, and you end up with two identical cells. There's a plant cell, which are more rectangular shaped. A wall would form down the middle, and you'd have two smaller cells.